Hello my dear students, this is Dr. 𝗲𝗵𝗵𝗺, your mentor of biochemistry. In this video, we are going to discuss the metabolism of branched chain amino acids. Right? So, let's start up with our today's video, branched chain amino acids. Now, whenever we talk about the amino acids, or whenever we talk about the metabolism, the structure of amino acids come to our mind, right? So what are amino acids? As we have discussed in our previous videos also, this is the generalized structure of the amino acids, and R varies. So classification as per R group, which we have already discussed. So, if we talk about the branched chain amino acids, first of all, they come under aliphatic group, or they are the aliphatic amino acids. And if we talk about the structure of these 20 standard amino acids, will be the same, this one. That is why we always study the metabolism as per the nitrogen skeleton and as per the carbon skeleton. So, if we talk about as per nitrogen skeleton, obviously since amino acids or the proteins are the wholly solely sources which actually give out urea, so it will give rise to the formation of urea. And if we talk about C skeleton, C will give rise to either glucose, so they will be glucogenic, or acetyl coenzyme A, so that they must be ketogenic. Now coming over to the branched chain amino acids, commonly we call them as BCAA. And BCAA play a very important role. If we talk about the function of branched chain amino acids, they play a very important role in the growth and the development of the child. Growth and development. And these branched chain amino acids are basically 3. So we can go through the mnemonic as leucine, isoleucine, and valine. So we can remember the mnemonic as LIV. Okay, so branched chain amino acids comprises of 3 amino acids, LIV or LIV: leucine, isoleucine, and valine. Now, if we talk about this structure, obviously they are having a branched chain, right? And if we talk about the nature of leucine, isoleucine as per the carbon skeleton, because nitrogen skeleton will remain the same for all the standard amino acids, valine. So, leucine is a ketogenic amino acid. Isoleucine is both, means glucogenic and ketogenic. And valine is a glucogenic amino acid. Now, if we talk about the structures, all are branched chain. So, if we talk about valine, generalized structure will be the same. So, I can say that this will be the same, NH2COOH. Now, instead of R group, it will have this type of structure. So, this is a branch chain, right? Then, if we talk about isoleucine, isoleucine will have the same structure. But the only difference will be here that it will have a iso group along with this. And if we talk about leucine, leucine is also going to have the same structure except here it will have this type of structure. So we can see all the valencies of carbon are met because we know carbon is tetravalent in nature. Okay, so this becomes the structure of valine, which is glucogenic. This becomes the structure of isoleucine, iso group is there, and this becomes the structure of leucine. So these are the 3 branched chain amino acids. Now coming over to this again, since I told you that the main function is one is a ketogenic amino acid, other is acting as both, and one is strictly a glucogenic amino acid. So directly we will see what is the metabolism involved here. So if we talk about the metabolism of branched chain amino acids, the 3 reactions or the 3 steps are similar to all the 3. Now, what are those 3 reactions? First is obviously transamination. We all know that transamination requires B6. So transamination mein enzyme is obviously aminotransferase or transaminases, but it will require PLP or B6. Second is, first they undergo transamination. The second similar reaction to all the 3 amino acids is oxidative decarboxylation. Now, oxidative decarboxylation, the coenzyme required is TPP or vitamin B1. Very important. And the third similar reaction to all the 3 amino acids is oxidative, or simply we can say FAD dependent. Dehydrogenation. And for this we require, since FAD is there, so we can simply say FAD or B. So these are the 3 similar steps to all the 3 amino acids, right? Now, this oxidative decarboxylation, as I have mentioned in my previous videos also, it always requires TPP as a coenzyme. And if we talk about the structure of this is again an example of multi-enzyme complex, just like PDH. And we can call it also as branched chain ketoacid decarboxylase or dehydrogenase. Branched chain ketoacid decarboxylase or dehydrogenase, or commonly we can call it as BCKD. So this BCKD is an example of multi-enzyme complex. Just like TPP present in PDH. Because again, if you remember, PDH is also an example of multi-enzyme complex, and the main coenzyme in PDH was B1 or TPP. Same is here. So if we talk about the composition of this, we can say that it again has E1, E2 and E3 as the enzymes which are helping in. When B1 is obviously decarboxylase, because here decarboxylation is occurring. E2 is transacylase and E3 is dehydrogenase. Okay, so I'll just mention like this. E1 is decarboxylase. This is transacylase. And E3 here is dehydrogenase. This is lipomide dehydrogenase, this is dihydrolipoyl, and this is branched chain ketoacid. It is branched chain ketoacid, this is dihydrolipoyl, and this is lipomide. And this is the composition of the enzymes. And if we talk about the coenzymes, it has B1 same, B2, B3, B5 and lipomide. B1 is our TPP, the main one. B2 is FAD. B3 is niacin or NAD and B5 is coenzyme A. Right, so this is the common reaction in all the three amino acids, and they are going to form their respective keto acids, and these respective keto acids will remove a molecule of this carbon dioxide, and then finally they will undergo different series of reactions. So this is how, I'll just show you the table that how the reaction goes. This is the complete table as you can see here. Right. What happens is leucine, isoleucine and valine. We can see here. So what is happening? Initially you can say that the transamination is occurring, which is obviously the first step. Okay, and transamination ke liye I can say simply, it's like aankhein band karke you have to just go for PLP. PLP ke saath it forms alpha-ketoacid. Alpha-keto because alpha-keto acids are produced, we know that. So this is caproic acid. Alpha-ketocaproate or caproate. This is alpha-ketomethyl-valeric acid and this is alpha-ketoisovaleric acid. So I will just write alpha-keto, so we all know this is isovalerate and this is methyl-valerate. As such, it is never asked in the exam, but yes, these three reactions are very important, which you should know. And for that, you can again go for the mnemonic. It's like TOD. TOD. TOD means T stands for transamination, O stands for oxidative decarboxylation, and D stands for dehydrogenation. So it's up to you. You can remember this. Then it undergoes. Second step is oxidative decarboxylation. So it is going to form isovalerial-CoA, alpha-methyl-butyryl-CoA and isobutyryl-CoA. Right? So this is your second common step, which is oxidative decarboxylation requiring TPP as a coenzyme. Then comes third reaction is, I told you, FAD dependent. Dehydrogenation. So if we talk about FAD dependent, you can see that HMG-CoA. Overall we can say. Then you can see propenyl coenzyme A, acetyl coenzyme A. This acetyl coenzyme A will obviously undergo TCA, or you can say propenyl coenzyme A forming succinyl coenzyme A. So wherever succinyl coenzyme A is being produced, we can say it is glucogenic. Whenever HMG-CoA is produced, we can call it as ketogenic. And that is why we can say that, if I have to summarize this, I can simply say that valine is strictly glucogenic in action. Always remember. Isoleucine is both. And if we talk about leucine, leucine is a potent ketogenic amino acid. Ketogenic. Because it produces HMG-CoA. HMG-CoA, we will be discussing this HMG-CoA and its importance in the metabolism of the amino acids. In the metabolism of the fatty acids, which is produced here with the help of amino acids. Okay, so this is the metabolism of branched chain amino acids. And this is very, very important. The structure of BCKD enzyme. Always remember, BCKD enzyme is an example of multi-enzyme complex. Multi-enzyme complex means it resembles pyruvate dehydrogenase in structure. How? It has three enzymes, E1, E2, E3, three subunits. E1 is having, again, E1 is having what? Jaise wahan pe it was PDH, here it will be decarboxylase. There it was again dihydrolipoyl. If you remember, it was trans-acetylase. Again, it is dihydrolipoyl trans-acetylase. And E3 is dehydrogenase. There also it was like that. And coenzymes B1, B2, B3, B5 and lipomide. And the most important one which you have to remember is TPP. And always keep one thing in mind, wherever oxidative decarboxylation is there, it's like by default TPP should come into your mind. And whenever decarboxylation is occurring, B6 should come into your mind. Okay. So this is how the metabolism of the branched chain amino acids takes place. Then third category which we study is, we studied what are the inborn errors of metabolism associated with this. So what are the inborn errors of branched chain amino acids? Right. So the inborn errors of branched chain amino acids, first one is Maple Syrup Urine Disease, and commonly we call it as MSUD, which stands for Maple Syrup Urine Disease. And second one is isovaleric acidemia. Right, this isovaleric acidemia is also known as sweaty feet odor. So, two diseases या two inborn errors of metabolism are associated with branched chain amino acids. Now, first of all, let's take up this first one, maple syrup urine disease. Now, if we talk about this disease, it is clear from the word itself, maple syrup, the urine gives the smell of burnt sugar. Okay, so first of all, let's take this. What is the deficient enzyme in this? So, if we talk about the deficient enzyme in MSUD, it is alpha-ketoacid dehydrogenase. So, the deficient enzyme is alpha-ketoacid dehydrogenase. Now, what are the clinical features associated with this? Mainly in the neonates. What happens here is, since branched chain amino acids are required for the growth and the development of the child, what will happen if the enzyme required to degrade this branched chain amino acids is absent? So, what will happen is, the main thing, if we talk about, the child will, suffer from hypoglycemia, hypotonia will be there, and he will be all the time, because he is not getting proper energy to work. So, he will be always lethargic, right? So, I can say lethargy, hypotonia or hypotonic, right? Because the muscles will not have that energy to perform any kind of function. So, what happens is, it will be difficult for the child to survive also, failure to thrive. Since the child is undergoing hypoglycemia, it might lead to convulsions. And the most important one is, the urine will give the smell of burnt sugar, as if some charring has taken place somewhere. Right? Urine gives the smell of burnt sugar. And if we talk about the biochemical investigations or the biochemical tests here, what will happen is, dinitrophenylhydrazine, which we call it as DNPH test, will be positive. And to our surprise, Rothra's test will also be positive. Rothra's test. Again, if we talk about, the biochemistry behind that or chemistry behind that, phenylhydrazine is there. So you can say it is going to form a violetish bluish type of complex. So DNPH or dinitrophenylhydrazine test. Nitrophenylhydrazine test and Rothra's test are positive in a child suffering from MSUD. And obviously, if we talk about the investigations, there will be increased concentration of the ketoacids and there will be increased concentration of the branched chain amino acids. This is very important. And if we talk about the treatment here, initially the treatment would be, because the child is suffering from the enzyme which is deficient to degrade BCAA, so treatment would be supplementation of vitamin B1 or TPP. Why? Because TPP is the one which is the major coenzyme required for utilizing branched chain amino acids. So this is the main treatment. Okay, so this is maple syrup urine disease, where the deficient enzyme is alpha-ketoacid dehydrogenase. Somewhere in some sources it is written decarboxylase also. So alpha-ketoacid decarboxylase, because this basically belongs to one complex. Okay, and these are the clinical features and this is the treatment. So this is the first disorder. Second is isovaleric acidemia. Now what happens in isovaleric acidemia? Isovaleric acidemia mein, this is also known as sweaty feet odor syndrome. First of all, if we talk about the deficient enzyme here, the deficient enzyme here is isovaleric dehydrogenase. And what happens is, here you can simply say that also known as sweaty feet odor. What happens is the urine, the body, whole body, in fact the cerumen wax also starts giving the smell that comes when our feet are full of sweat. So that stingy type of smell will come from all over the body. So be it like urine, be it like your wax, everywhere it will give this smell, sweaty feet odor. Even cerumen wax also will give this type of smell. Right? So these are the two main disorders which are related to branched chain amino acids, which are very, very important for the proper growth and development of the child. Clear? So this is all about this video. I hope you have understood, because the main thing which you have to remember here is, I told you starting say in all the videos you will see, this is how we differentiate. Ki whenever any kind of amino aciduria is involved, it is related to amino acid metabolism. Okay, when a child is suffering from phenylketonuria, the moment you open the diaper, it will give a musty odor. Here you can say the urine will give a kind of maple syrup odor. So these are the distinguishing or the diagnostic criteria of differentiating different type of amino acidurias with the metabolism of the carbs and lipids. Right? So this is all for this video. Thank you so much.